Hi, I'm Jilian Melamed, and I'm a research assistant professor at the University of Pennsylvania in the Institute for RNA Innovation. And today I'm excited to share with you how we can use mRNA-lipid nanoparticle vaccines to develop new treatments for autoimmune disease. So many of you may have heard of autoimmune diseases before, but for those of you who haven't, autoimmune diseases are when the immune system attacks your own body because it thinks it's a foreign pathogen that it should destroy. Autoimmune diseases are incredibly diverse and can affect nearly every tissue in the body. In diseases such as type 1 diabetes, the immune system attacks and destroys the insulin-producing beta cells in the pancreas. In multiple sclerosis, the immune system attacks myelin in the brain. So you get the idea. Autoimmune diseases can affect virtually every organ or tissue in your body. And in fact, there are over 100 known autoimmune diseases, and unfortunately, they're on the rise globally. They affect roughly 5 to 10% of the global population, and they disproportionately affect women, though we're not sure why. There are no known true causes of autoimmune disease, and there are a number of key factors that are important and we understand play a role, such as your genetics, your environmental conditions, and your unique immune system. But we haven't pinpointed a perfect cause for every autoimmune disease. And what's really most unfortunate is that there are no cures available for these diseases. A lot of the available treatments will focus on managing symptoms. For example, patients with rheumatoid arthritis and lupus rely on a lot of pain management. And there are lifestyle modifications that could be made, particularly in the case of type 1 diabetes or celiac disease, where you can change your diet. But for severe autoimmune diseases, in particular lupus and rheumatoid arthritis, some of the treatments are just to broadly suppress the immune system to prevent it from attacking itself. And this is effective in that it can keep unwanted inflammation in check a bit, but it also leaves these patients vulnerable to infections and even cancer. So, what we really need are new treatments that can suppress the immune system in an antigen-specific manner, meaning we're only targeting the cells that are driving the specific autoimmune disease forward. So your immune system can normally recognize itself and avoids attacking itself. And this is called self-tolerance. Self-tolerance is maintained through two mechanisms, one of which is central tolerance. And this is how your immune cells undergo primary training. Your T cells develop in the thymus, B cells develop in the bone marrow. And during this process, as T cells and B cells are developing, your body keeps cells that respond to foreign pathogens and delete cells that are self-reactive or autoreactive. And these are the cells that might see insulin and mount an immune response, or see myelin and attack it. The other mechanism by which self-tolerance is maintained is in the periphery throughout the body. And we can think of this as secondary immune surveillance throughout the tissues. And throughout your body, you have effector T cells, which hopefully are only responding to pathogens like viruses and bacteria. But you also have regulatory T cells, and these are the cells that are responsible for suppressing unwanted inflammation. And in doing so, they maintain self-tolerance by stopping effector T cells from attacking your own tissues. So when autoimmune diseases are occurring, it's because self-tolerance breaks down. And to initiate this, autoreactive cells can escape selection in the thymus and the bone marrow. And in many autoimmune diseases, regulatory T cells or Tregs throughout the body are defective. They can be reduced in numbers, their suppressive functions can be impaired, they can be worse at getting into cells where autoimmunity is, or tissues where autoimmunity is taking place. Um, so my research goal is to overcome these challenges and restore tolerance in autoimmune disease. And we can use vaccines to do this. This is a very different class of vaccine than you're used to hearing about, but we call them tolerizing vaccines, and I'll walk through with you how they work. To understand how a tolerizing vaccine works, I like to compare them to an infectious disease vaccine. So, to develop a vaccine for an infectious disease, you want to deliver whatever your antigen of interest is, a piece of a virus that your immune system can recognize, such as our COVID spike protein. And we want to deliver that antigen to immune cells with lots of inflammatory cues. And this is called an adjuvant. So we take our COVID spike protein, we deliver it with inflammatory cues to antigen presenting cells. And those antigen presenting cells are going to break down that protein and present pieces of it on MHC complexes. And this is how antigen presenting cells interact with T cells. So your antigen of interest is presented on MHC, which then engages with T cell receptors on T cells. And in the presence of inflammatory signaling, we get differentiation of effector T cells. And one cell type that we're going to talk about that are induced by vaccines are T follicular helper cells, which produce inflammatory signals that alert the body that there's a virus, there's something dangerous present. And these cells help make germinal center B cells, which are the cells that make antibodies that help your body clear infection. In a tolerizing vaccine, we want to essentially deliver an antigen that is already present in the body, so like insulin for type 1 diabetes or a myelin protein for multiple sclerosis. And we want to do that with no inflammation at all. So in this case, we're going to deliver a self antigen without any adjuvant. And in doing so, we can elicit an antigen-specific regulatory T cell response that will hopefully be sufficient to counteract the effector autoreactive T cells in the body. So in this way, we can elicit antigen-specific suppression and get around the need for broad immunosuppression to treat autoimmunity. The technology we use in my lab is the mRNA-lipid nanoparticle vaccines. And today I'll tell you about how we can use them to make tolerizing vaccines. So first, you may be wondering, what are mRNA-lipid nanoparticles and how do they work? Well, they have two important components. The first is the mRNA, and mRNA is genetic material that we can design to make any protein we want. mRNA is a safe therapeutic. It cannot enter the nucleus. It cannot change your DNA. And another benefit of mRNA is that it's transient. Its effects are short term. So it can produce the desired protein for a short period of time to enable antigen presentation and engagement with T cells. And then the mRNA is degraded and the protein is no longer expressed. And the LNP is essentially a delivery vehicle that's designed to encapsulate and protect the mRNA cargo on its way to immune cells to do its job. mRNA is a very sensitive molecule and it's easily degraded by enzymes that are present throughout our bodies, on our hands, and on all of the surfaces. So in order to deliver it effectively, it needs to be encapsulated in a delivery vehicle that mimics the body's cells. And we do that with lipid nanoparticles, which are essentially just tiny balls of fat containing a mixture of lipids that we'll talk about in a few slides. And in this way, lipid nanoparticles actually look like tiny pieces of your own cells. So your body perceives them as being safe. What's great about them is that they are really good at delivering mRNA. But unfortunately, for applications in autoimmune disease, they're highly inflammatory. So that makes them great for infectious disease vaccines and challenging for vaccines for autoimmune disease. And this inflammatory nature of LNPs is called adjuvant activity, and it is a natural property of the lipid nanoparticle itself. So on the left of the slide, I have a little reminder that in a vaccine, you have a naive T cell. A vaccine provides lots of inflammatory cues that induce T follicular helper cell differentiation. This produces inflammatory cytokines and ultimately antibodies. And I have some friends who did a really cool experiment to show that this is a property of the LNP specifically, because they showed that when you take a traditional protein vaccine and you spike an LNP into it, you get the same quality of immune response. So here we're looking at an experiment where they looked at the number of activated T cells, and they did this in naive mice or mice that haven't been treated with anything, in mice that received a protein vaccine plus Adavax, which is a traditional vaccine adjuvant. But most importantly, they compared this protein plus LNP to mRNA encapsulated inside an LNP. And what I want to highlight for you is that the protein plus LNP and the mRNA plus LNP groups contain the same number of activated CD4s. So this is great for infectious disease, but really challenging for autoimmunity. And that's where I come in. My research focuses on engineering LNPs that are good at delivering mRNA to immune cells, but without the pro-inflammatory adjuvant activity. And we can do this because the immunological properties of LNPs are dictated by the lipids that make them up. This is a cartoon representing our best guess at what a lipid nanoparticle currently looks like, though that's under debate. And lipid nanoparticles contain four lipid components plus the mRNA encoding our protein of interest. So these are the ionizable lipid, some helper phospholipids, cholesterol, and PEG lipids. For the purposes of this talk, the cholesterol and the PEG lipids primarily serve to impart stability to the lipid nanoparticle. And for this talk, we're going to focus on the ionizable lipids and the helper lipids and describe how they can be used to develop LNPs that deliver mRNA to immune cells without inflammatory adjuvant activity. So you may be wondering what I mean by ionizable lipid, and they are the most important component of the LNP. And this is because when LNPs are administered to the body, they need to be taken up by target immune cells. So here I have a little schematic of an immune cell taking up our little orange colored LNPs. So LNPs are administered to the body. Immune cells take up LNPs by a process called endocytosis, where they enter these little vesicles called endosomes. And once the LNPs enter the endosome, the pH drops. Endosomal pH is lower than that of physiologic pH. Physiologic pH is around 7.4, but once LNPs enter the endosomes, the pH drops to around 5.5, and the ionizable lipid ionizes. And that just means it becomes positively charged. And now you have all of this positively charged lipid in an endosome made up of negatively charged lipids. And the ionizable lipid is going to interact with the endosomal lipids. And this causes the mRNA to be released inside of the cell so that it can make whatever protein you're interested in making. So without the ionizable lipid, you do not get mRNA released inside cells. So they're incredibly important. And helper lipids have been conventionally thought to simply imbue stability to lipid nanoparticles. But more recent research has shown how helper lipids with different charges can shift LNP delivery to different cell types and organs within the body. So in this instance, I'm going to show you how we measure this. And this is done, we make LNPs encoding a reporter protein, and that just means anything that's easy to measure. So in this experiment that I'm about to show you, we made LNPs with mRNA encoding firefly luciferase. And yes, I mean firefly luciferase, like the protein in fireflies that makes them glow. So we make LNPs encoding firefly luciferase, we inject them intravenously to mice, and then 6 hours later, we use an instrument called an in vivo imaging system to measure the amount of light produced. So if there is more protein produced by delivering the LNP, we'll measure more light. So here I'm showing you results from this experiment. If we look at the top set of images, the liver, lungs, and spleen from mice that were treated with LNPs containing neutrally charged helper lipid DSPC, we see that most of the protein expression, most of the light that we're measuring is within the liver. And this is true for almost every LNP, goes to the liver, delivers protein really well to the liver following intravenous administration. But those aren't the cells that we want to hit for a tolerizing vaccine. We want to hit immune cells present in the spleen. So in this case, we formulated LNPs containing a negatively charged helper lipid called DOPS. Made these with our same luciferase mRNA, delivered them to mice. And in this bottom set of images, you can see how we have less light produced in the liver and more light produced in the spleen. And the immune cells we want to hit are located in the spleen. So then my next question was, what is the effect of these anionic lipids on adjuvant activity? Do they make the LNPs more inflammatory or less inflammatory? And to understand that, first we need to understand how immune cells sense and transmit adjuvant activity. So here I have our antigen presenting cell friend and our effector T cell friend returning. And in order to get an effector T cell response, you need three separate signals. The first is antigen presentation, and we control this with the mRNA cargo that we design encoding whatever our antigen of interest is. It's delivered to the APCs, and then it's processed and presented on MHC, where it can engage with T cell receptors on T cells. The second signal we need for good adjuvant activity is co-stimulation. And this is mediated by surface molecules on APCs like CD80 and CD86. So more CD80 and CD86 expression, more adjuvant activity. And the result of all of this is the production of inflammatory cytokines. So these are extracellular cues that cells can transmit to each other to alert the body whether danger is present, whether it needs to initiate tissue repair, to initiate all kinds of immunological processes. So when we have all three of these signals together, antigen presentation, co-stimulation, and inflammatory cytokines, we get an effector T cell response. This T cell will recognize this antigen and attack and destroy it. However, if we only have one signal present, just antigen presentation, we can get a regulatory T cell response. So in order to do this, we need a lipid nanoparticle that is non-inflammatory, that doesn't cause upregulation of CD80 and CD86, and that doesn't lead to the production of cytokines. So the question I asked was, which LNPs are the best at reducing inflammatory cytokines? Typical LNPs are very pro-inflammatory and they cause the body to make lots of IL-6. And this leads to robust differentiation of T follicular helper cells. Again, great for infectious diseases, bad for autoimmunity. We want to reduce that. So what I did was I tested a library of 40 different lipid nanoparticles. And I looked at four different ionizable lipids that are shown here on the vertical axis of what is eventually going to be my heat map. And I tested two different vaccine lipids, SM-102 and ALC-315. You might have heard of these before. These are the ionizable lipids that are in the Moderna vaccine and the Pfizer vaccine. So SM-102 is the Moderna vaccine, ALC-315 is the Pfizer vaccine. And I also tested two different ionizable lipids that are more commonly used for protein replacement therapy. MC3 down here at the bottom was actually the first ionizable lipid to be FDA approved for RNA delivery. And in the case of MC3, it's siRNA delivery to the liver. And 306-OI10 is a good protein replacement lipid that I helped develop as part of a previous lab. And what we really like about it is that it's fairly non-inflammatory. So I took each of these four ionizable lipids and I paired them with one of three anionic lipids. The anionic lipids I chose are called DOPS, BMP, and DOPG. You'll also see a bunch of tick marks across the horizontal axis, and those are just different fractions of anionic lipid within the LNP. I tested low fractions of anionic lipid up to some higher concentrations of anionic lipid in LNPs. So these LNPs were all formulated with mRNA and delivered to mice. And the readout we're looking at here is the amount of serum IL-6 that is produced. So we can collect blood from the mice, measure the amount of inflammatory cytokine, and that tells us about the adjuvant activity of the LNPs. So here we're looking at serum levels of IL-6 in picograms per milliliter. This heat map is going to populate with a bunch of colored boxes, and boxes that are more purple mean that there's less IL-6 present, and boxes that are more green to yellow mean there's lots of IL-6. The first group we looked at is our control group. So this was mice that were not treated with LNPs at all. As you can see, the boxes are all purple. We don't have a lot of IL-6 being produced. The second group we looked at was LNPs that didn't contain an anionic lipid. So these are more traditional four-component lipid nanoparticles that are used for vaccines and protein replacement therapies currently. And what you can see, particularly for our vaccine lipids, SM-102 and ALC-315, we're getting lots of serum IL-6, exactly as you would expect for an LNP that is good as a vaccine. For MC3, we're seeing a fair amount of IL-6 produced, but for the ionizable lipid 306-OI10, we're not seeing very much. And that's exactly how we know that all of these lipid nanoparticles should behave. If we take a look at our first anionic lipid, DOPS, you do not have to memorize the color of every one of these boxes. The only thing I want you to take away from this is that adding the anionic lipid reduces the amount of IL-6 that's being produced. So you're starting to see more blue and purple bars. If we take a look at our next anionic lipid, BMP, we're seeing an even further reduction in the amount of IL-6 produced. So these are LNPs with even lower adjuvant activity. And finally, if we look at the anionic lipid DOPG, this is where we're seeing the greatest reduction in adjuvant activity. So we thought that some of these LNPs might be good as tolerizing vaccines to deliver mRNA encoding autoantigens without any inflammatory cues and get an antigen-specific Treg response and halt autoimmune destruction of tissue. So of course, our next question was, well, if we take a couple of these LNPs, are these going to be good enough to induce tolerance? So we turned to two different mouse models of autoimmune disease. And in this case, we were interested in looking at multiple sclerosis and rheumatoid arthritis. These are two very different autoimmune diseases that are common and affect different parts of the body. So first, let's take a look at what we did in multiple sclerosis. For those of you who haven't heard of multiple sclerosis before, it is an autoimmune disease that affects the central nervous system. Your nerve cells are typically insulated with a protective myelin coating, and this insulates your nerve cells to increase the speed of action potential travel across axons. So all of this means is your brain needs myelin to transmit signals quickly. However, patients who suffer from multiple sclerosis have autoimmune destruction of those myelin sheaths. So in the cartoon on the slide, I'm showing you a nice picture of what healthy myelin looks like. But in multiple sclerosis, we get myelin destruction. So we're losing all of this insulation across the length of our nerve cells. So this affects the quality of signals that your central nervous system can transmit and will eventually lead to nerve damage. So in patients, this looks like difficulty walking, vision problems, muscle stiffness, numbness or weakness in the arms and legs. And there are no cures for these patients. These patients receive systemic immunosuppressants, so they are globally immunosuppressed. And in many cases, that's not sufficient to prevent disease progression anyway. So many scientists have been trying to understand multiple sclerosis and identify potential cures. So what we do know is that there are a couple of proteins in myelin that the immune system is sort of latching onto as autoantigens that propagate the disease. And I have a couple of them listed at the bottom of the slide here, but they include MBP, PLP, and the protein we're going to focus on for this portion of the talk, myelin-associated basic oligodendrocyte glycoprotein, and we'll just call it MOG. So what we wanted to know is if we took mRNA encoding the MOG protein and we encapsulated it in our non-inflammatory lipid nanoparticles, is this sufficient to reduce disease progression in a mouse model of multiple sclerosis? And this mouse model has a really long name. It is experimental autoimmune encephalomyelitis, or we just call it EAE for short. And to induce this mouse model, we inject mice with myelin proteins emulsified in adjuvant, so with lots of inflammatory signals on days 1 and 2 of the experiment. And then over the course of about a month, the mice begin to develop symptoms of multiple sclerosis. On days 7 and 10 of our experiment, we take our non-inflammatory LNP with mRNA encoding the MOG protein, and we deliver that to the mice. And then for the remaining 15 days of the experiment, we monitor them for the onset of paralysis, and we're able to look at myelin integrity in the brain and spinal cord at the end of the experiment. So here we're looking at what a typical course of EAE looks like, and these mice show progressive paralysis and recovery. This graph is showing you mice that have been induced for EAE but have not received any intervention. So on the y-axis, we're looking at EAE severity, and this is a severity of the paralysis that the mice are experiencing. EAE begins in the tail and in the hind limbs. So mice that experience a limp tail receive a score of 0.5. And as the paralysis progresses, the EAE score increases. So here we see that mice don't start to experience symptoms until around day 12, and then they will rapidly get worse in this initial phase. And then mice, unlike humans, are really good at remyelinating. So this is followed by a period of recovery. And over the course of this entire experiment, it's really important that our scoring is performed by observers who don't know what the mice are treated with so that we can avoid any bias in our scoring measurements. So here we have 3 different treatment groups. Our PBS control is mice that were injected with saline. We know that this shouldn't do anything. We have this OVA LNP group, and this is mice that were treated with an LNP encoding an irrelevant protein, in this case, ovalbumin. So this group tracks right along with PBS in these studies, and that's really important because it tells us that any therapeutic effects we might see are due to the mRNA and not just the LNP. And what we were really excited to see was that if we treat mice with LNPs encoding the MOG protein, we see a pretty drastic reduction in disease severity. So this was very exciting. So then we took some mice and we sacrificed them and looked at their spinal cords so that we could see what the myelin looked like. And here we're looking at sections of mouse spinal cord using a technique called immunohistochemistry. And this allows us to visualize the structure of tissues. And the brown you're looking at here is myelin basic protein. So anything that's brown is myelin. And you can see that if we look at our MOG LNPs first, there's lots of myelin present. It is showing nice structure, and there aren't any disruptions in the myelin. However, if we look at our PBS and OVA-treated controls, we can see lots of immune cell infiltrate, and that's indicated in blue, where we have these dense clusters of cells. The blue shows the cell nucleus. And throughout the white matter for the PBS and OVA LNP treated mice, we have lots of immune cells that are infiltrating the myelin and causing it to degrade. If you again compare that to the MOG group, the myelin remains intact. So this is indicating that our treatment prevents the destruction of myelin in this model, which was really exciting. Of course, in this model, we initially treated mice with mRNA-LNPs before symptom onset, so days 7 and 10. But that's not realistic when you have patients coming in. Patients coming in are only going to see a doctor when symptoms start to appear. So we also wanted to see if our LNPs are effective when administered in a therapeutically relevant setting once mice start scoring a 1 or a 2. So in this case, we took mice and treated them once they started showing symptoms of disease. And in fact, the results were maybe even a little more impressive, where the MOG LNPs were capable of reducing EAE severity even after symptoms had already taken place. So this is very exciting. And we're actually hopeful that we'll be able to further develop this platform into a treatment for multiple sclerosis. Um, However, this particular multiple sclerosis model was reliant on a single antigen. That means it was induced using the MOG protein and it was treated using the MOG protein. So this is a really simple disease model, and while it's effective for validating an initial platform, many human autoimmune diseases are a lot more complicated than that. They're caused by multiple antigens. So for this, we turned to collaborators and their model of rheumatoid arthritis to ask if our tolerogenic LNPs were effective at treating a more complicated disease model. So for this, we worked with collaborators out at Utrecht University, Dr. Femke Brouwer and Dr. Naomi Ben. And they have identified a really important autoantigen implicated in rheumatoid arthritis. And it's called MB29A. And MB29A is a peptide derived from a heat shock protein. Heat shock proteins are expressed at higher levels during inflammation. So in doing that, this vaccine will be very specific for autoimmune inflammatory arthritis. So we wanted to know if our mRNA-LNPs can treat a mouse model of rheumatoid arthritis. And in this case, the mouse model is induced with a mixture of human proteoglycans. So that means that lots of complex antigens are present and causing autoimmunity in this model. We treated the mice with lipid nanoparticles containing mRNA for our MB29A protein, and then the mice were scored for arthritis for 50 days. You may be wondering what arthritis looks like in a mouse, and the way it's measured is based on paw appearance. So if we look at the paw picture all the way to the left, this is what a score of 0 looks like. It's a nice, healthy mouse paw. But as these mice develop arthritis, you might see some toes start to swell in a score of 1 or 2. As it gets worse, you start to see swelling in the ankle, and at a score of 4, the paw is completely swollen. So the way the scoring works in this model, mice have 4 paws. Each of the 4 paws is scored, and the scores are added together for a maximum score of 16. So here again, we have our arthritis score on the y-axis. We have days after the disease induction on the x-axis. And if you look at mice that were treated with our irrelevant OVA LNPs, the arthritis becomes progressively worse. But if you look at mice that received our MB29A encoding LNPs, the arthritis didn't progress to nearly the same extent. So we were really encouraged by all of these early experiments and early results. And we hope that we've demonstrated and convinced you that mRNA lipid nanoparticles are a great platform that can be easily modulated to induce antigen-specific tolerance as a treatment for a variety of autoimmune diseases. We've found that whether or not the LNPs will induce a tolerogenic response depends on their lipid composition. And two of the most important lipids for that were the ionizable lipid and the anionic helper lipids. And another thing that we've found along the way is that this isn't necessarily a one-size-fits-all therapy. The mRNA, the lipid nanoparticle, and how you deliver it to mice and eventually to humans does have to be tailored for many different autoimmune diseases. So I like to end my presentations by thanking everybody who contributed to the work. Here's a picture of the Weissman lab where I work at Penn. Um, the names listed here are folks on my team who contributed to getting all of these experiments done. Of course, our collaborators out at Utrecht University. We have collaborators at Cincinnati Children's Hospital who have been so important for getting all of this work done. And I'd also like to thank the people who provided the funding to do the work. And thank you, of course, to all of you for listening.